what I want to talk about really briefly is the very simplest, which is perhaps a hint as where this stuff started. The simplest form is bacterial, and you might call it sex. It's a little, there was the way it works is the following. They pick up DNA fragments from other, even dead bacteria. And so, for example, transmission of antibiotic resistance via plasmids. These are small chromosomes. So if you remember from Bio 111, there was P-glow. There was that plasmid. If you put it in bacteria, it had new features, including antibiotic resistance. Turns out P-glow was derived by human modification from something that naturally evolved. There were plasmids in E. coli that allowed them to pass around antibiotic resistance. And it's a problem, if any of you work in hospitals, you know about this, that you end up with situations where bacteria swap genes like this. So you have a bacteria that's resistant to tetracycline, another one that's resistant to ampicillin. And they get together, and you now end up with some superbug that's resistant to both. And the way they do it is by passing around these plasmids. And it turns out bacteria have mechanisms for slurping up DNA from the environment and just kind of using it. And sometimes it kills them. Oh, well, they're dead. There's lots more bacteria. Sometimes it helps them. So much the better. Right? And there was a talk I saw someone once about this that said, bacterial sex, why sex with dead things is better than no sex at all. And so that was that's basically how it works for them. And it is quite successful. It's not a full-scale cross. It's not like you get half your genes from your mom and half your genes from your dad. It's like you pick up a piece of DNA and you use it. Right? But it's sort of on the way towards more complicated sexual reproduction. All right. What I want to spend the rest of the day talking about is in eukaryotes. Plants, animals, and fungi. It involves meiosis and alternating between haploid. So you can use these terms a lot. Haploid is called sometimes N or 1N, one copy of each gene. So that would be big A or little a. And diploid, 2N, two copies of each gene. And they can have genotypes big A, little a, little a, for example. Right? And we spent a lot of time in 111 talking about the genetics of diploids. The only time we ever ran into haploids, to the extent that we even thought about them, were eggs and sperm. It's going to turn out they play a much bigger role in things than you might expect. And we're going to talk about that in a